Hello and welcome to this webinar on best practices for phenotypic drug discovery. This has been created by the Best Practices in Medicinal Chemistry Working Group for the European Federation for Medicinal Chemistry. And as you can see on the slide, this is a group of people from different pharmaceutical companies as well as academic institutions. So what we would like to do today is to guide you through the basics and general aspects of phenotypic drug discovery and share some best practices along with that. Associated to this webinar, you will also find a presentation containing 2 case studies of phenotypic screening projects that will highlight and exemplify several aspects out of this main presentation. First, we are going to introduce phenotypic drug discovery and its key principles. The presentation will then follow the flow of a phenotypic drug discovery approach from the assay that is used, the screening library aspects, onto the hits and what techniques are used to validate them. Finally, a main aspect of phenotypic drug discovery is the target elucidation, and we will summarize techniques and technology used to deconvolute and identify the targets of phenotypic drug discovery programs. Now, what is phenotypic drug discovery? Let's define a phenotype in the context of drug discovery first. An organism's phenotype is a result of 2 basic factors: the expression of its genetic code, its genotype, and the influence of environmental factors. And overall, a phenotype can be observed and quantified. This covers the organism's morphology or physical form and structure, its developmental processes, its biochemical and physiological properties, its behavior, and the products of this behavior. So the concept of phenotypic drug discovery implies running an unbiased screen to observe this quantifiable phenotypic endpoint and to find compounds that produce this desired phenotype. However, this is done without prior knowledge of the targets. So it aims at modulating disease-relevant phenotypes, but without making assumptions about a specific target or a mode of action. Often that is also called mechanism of action. This mode of action will only be elucidated or deconvoluted much later. This is in contrast to a target-based screen, where you would look for a mode of action first and then just screen on that target, and only later, after optimization, one would test the modality, the compound in an organism. In this webinar, we will focus on small molecules, while a phenotypic drug could also be a peptide or a RNAi. In recent years, there has been quite some discussion around a comeback of phenotypic screening and its advantages and disadvantages. If you look at new chemical entities, in general just first-in-class small molecules in these graphs, approved over a certain time span, you will see that the ones discovered through phenotypic screening, shown in blue, had quite an impact over a certain time before 2008. And this fact has been cited a lot as a basis for the renaissance of phenotypic drug discovery. On the other hand, an analysis of a longer time span, including later years, shown on the right, revealed that phenotypic drug discovery has had a much lower impact overall during that referenced time span, in terms of approved small molecule first-in-class drugs. Now, there are several reasons to explain the shift over the years. Many technologies have been developed over the last years that strongly support target-based drug discovery, spanning from, for example, genomic tools for target hypotheses and identification to biophysical and computational methods to support knowledge-based approaches, etc. Also, the impact that multiple approved kinase inhibitor had, for example, in the oncology area, All that shifts the numbers towards target-based approaches. Nevertheless, phenotypic drug discovery has had an impact and led to approved therapies that contribute to the well-being of humankind. There is no better than the other. We will present in this webinar not just the positive aspects of phenotypic screening, but also illustrate the challenges and uncertainties that are associated with it, to help set realistic expectations with regard to its benefits. In order to be successful, the research in phenotypic approaches need to grow beyond the current state of the art and invest in new technologies. The choice should not be between phenotypic screening and target-based drug discovery. Both approaches can actually complement each other. To give you a few examples of drugs that have been previously approved or are prior approval that originate from a phenotypic screening approach, we have listed just a few, including their therapeutic area and a few details on their discovery. The three molecules on the left, pirfenidone, ranolazine, and levetiracetam, applied in respiratory, cardiovascular, and CNS diseases respectively, have been discovered from starting points found through disease-specific animal models. They represent more the historic examples of successful phenotypic approaches. Lubiprostone is an example for a compound that is related to an endogenous prostaglandin ligand, although the now postulated mode of action is actually to modulate chloride channels and increase fluid secretion into the intestinal lumen. Fulvestrant is an example for a compound that had originally been discovered in a target-based approach for the discovery of a pure estrogen receptor antagonist, but was then later found to be impacting the receptor's control of gene transcription due to induction of conformational changes and is now used in cancer therapy. A very recent example is ridasplam, a compound for the treatment of spinal muscular atrophy, for which the discovery is highlighted in parts in the case study section of this webinar. It really shows the application and impact of advanced methodologies in phenotypic drug discovery. So what are the potential advantages and opportunities of phenotypic drug discovery? The key aspect of employing a phenotypic approach is the possibility to capture a disease-relevant mode of action without full mechanistic understanding and knowledge of a target, and to do this in a physiological relevant context. Therefore, you can exploit a disease phenotype to discover novel treatments for diseases for which even the root cause is unknown. Sometimes this is referred to as forward pharmacology. It sounds very straightforward and simple here, but is actually very complicated, as you will see in the course of the webinar. And just to clarify, phenotypic drug discovery should not be regarded as an alternative screening technology or as an easy fix to the challenges of clinical attrition rates. One of the other reasons to do this is the desire and the potential that the translation into a meaningful in vivo readout and ultimately into a clinical outcome is higher. This is based on the fact that the systems that are used are much different than just isolated targets in form of, for example, enzymes or overexpressed targets in simple cell systems, which are often used in target-based drug discovery. In the course of phenotypic projects, it is then possible to discover new targets or also new physiology for known targets. Additional benefits are also highlighted in this graph. With phenotypic approaches, it is possible to identify multiple disease-relevant effects, not just an effect via one target, written here as molecular mechanism of action. Each mechanism could have its own efficacy versus safety profile. The potential to find desirable so-called polypharmacology is a key benefit of phenotypic approaches. In contrast, in a target-based approach, shown in the box on the left, the efficacy versus safety profile will be fixed or mostly fixed if there's an intrinsic safety liability with that target. But the safety risks with unknown targets also need to be carefully evaluated. That brings us to the challenges of phenotypic drug discovery and to the next slide. To perform a screen in a system that has a high biological relevance, and we will get to that topic in a second in more detail, it comes with a higher complexity and often it is more expensive. One of the major hurdles after a phenotypic screen is a more challenging hit validation, resulting from using these more complex systems. And as we have learned on the previous slide, the more options you will get, also the more unwanted mechanisms might be included and need triaging. Logically, the compound optimization in SER can be more complex and data-rich than in a target-based program. Especially with new and unknown mechanisms, as mentioned, safety risks have to be assessed. To achieve a needed early de-risking, the primary assay needs to be carefully selected and multiple counter screens and orthogonal assays need to be in place. Ideally, you would look into an early in vivo confirmation and the use of high-value biomarkers, and also perform early safety profiling. After we have now introduced the aforementioned general aspects of phenotypic screening, let's stop here and define a set of key questions that we think are crucial for a successful phenotypic screen. We will then go into each of these topics in more detail. First of all, in what system do you want to screen in? And that comes down to a question of throughput versus disease relevance. Second, which compound should we screen? Can we go for large compound sets, or are we restricted in throughput? And do we maybe include specific compounds that we already have certain knowledge about? We will discuss this. Third, when we get results, how can we actually validate those hits? We need proper flow charts to ensure filtering out relevant hits. And last, how can we identify the molecular target of these compounds? We will discuss several techniques to identify the mechanism of action. Starting with the assay. Which assay system you can use is of course influenced in general by the biological question that you want to answer. For example, if you want to impact tumor growth, you would use a tumor cell. Similar as in target-based drug discovery, the complexity of the system defines your throughput. But here, using systems that are more complex and that are closer to the true disease setting, the aim is also to offer a higher biological relevance and better translatability. As such, looking at the table here, simple cell systems on the left are easier to handle, cheaper, and often more readily available than, for example, patient-derived primary cells listed on the far right. But these might actually, on the other hand, give you that aforementioned higher biological relevance. The readout, however, might come with more variability. Regarding throughput, one needs to consider the following, going again from left to right. Now in the assay platform row, it is easier to achieve a higher throughput with a simple DNA reporter-based readout versus a more complicated analysis using, for instance, live cell microscopy and sophisticated image analysis. So what we have just discussed here highlights that several aspects need to be factored in when deciding for the right assay system for your phenotypic project. As important as the right assay system and very much connected to that topic is the question of assay activation or stimulation. And after that, the nature and the relevance of the readout. We are going to look at both on the two following slides. Aside from the fact that some assay systems used for phenotypic screening might not need a direct stimulus to produce a disease phenotype, like some patient-derived cells or induced pluripotent stem cell-derived cells with disease-causing gene mutations, your assay system will likely need a stimulus. While it is clear that this needs some knowledge about the pathway that might be causative for the phenotype, it is advised to use a stimulus that is close to the disease-related root cause. If not, you may create a risk of missing relevant mechanisms while capturing some that would not translate to patients. This is shown here in the graph A. Consequently, a stimulus that is far downstream in an interrogated signaling pathway would not activate upstream elements, which would then run the risk of being not captured in the screen. This is visualized in graph B. Also of importance to limit impacts of false positives later in the readout is to test your assay system with unspecific activators, like for example, electron transport chain inhibitors, to ensure that the system does not react to those stimuli in a way that resembles the applied assay stimulus and that would then appear as false positives. Speaking of readout, this also has great relevance for the success of a phenotypic screen. The more relevant the readout is, the higher the likelihood of translating to clinical efficacy, as the assay readout moves closer to the clinical endpoint. This is further illustrated by the picture on the right, showing that a gene expression might be too far upstream and would not capture a modulator of post-translational modification, for example. It is sure possible to use gene expression readouts, and this can be successful. However, considering a more functional readout or even a macrophysical is ideal. Another point to make regarding readout is to acquire multiple data points at once if possible. This is often referred to as high content analysis. In a cell-based screen, this could be, for example, cell number, morphology endpoints, and impacts on mitochondrial fitness using certain markers. In summary, the more relevant the readout is, the more mechanisms modulating this phenotype are captured, and the higher the translatability to patients should be. With the fundamentals in assay design discussed, we would now look into which compounds to screen. What are the right compound design criteria that will support generation of high-quality hits and lower false positive rates? Wherever possible, it is advised to use compounds that fulfill general physicochemical quality requirements. This is more and more the case in compound collections around the world. It also depends on the assay type. For example, permeable compounds are advised when you envision modulating an intracellular target, and if you do not want to rely on active uptake. Solubility and aggregation or instability can also lead to false positive and false negatives, respectively. Regarding false positives, looking into removing compounds that are considered troublemakers, such as frequent hitters, can help reducing false positives. In case a proof of concept experiment in an in vivo model is planned and the in vivo model is available, the use of screening compounds that already possess good drug-like properties enables a rapid progress. Also, if the throughput of the assay is very limited, it makes sense to start with a well-cured compound set. Thereby, you enable any downstream experiments without the need for a lengthy compound optimization. This leads us to a very general remark that is also true in target-based drug discovery programs, that the format and the cost of the screening assay, as we have discussed in the previous slides, often determines how many compounds can be screened and how much effort needs to be put into designing that screening set. This also applies to the diversity of the compounds within the set, which brings us to the next point. The key question that you need to answer already during assay design and before starting your screen is, what is the desired outcome of the screen? The first scenario that we're going to look at to answer this is the one where you would want to find novel biology for known targets and their compounds. This concept is often referred to as biological diversity and also well known as using an annotated compound library, meaning that a priori the targets hit by the compounds that are used are known. The limitations to this approach are somewhat obvious. In principle, you restrict yourself to known mechanism of action, and you will mostly only find new biology for these targets. Another drawback that complicates triaging is that often the annotation is incomplete, and although you wanted to avoid doubts about the targets by using an annotated library, you will actually have to look into verifying your target hypothesis afterwards again. So when compiling such an annotated compound set, you can rely on commercial sources or internal knowledge about the targets of these compounds. It is smart to include several compounds annotated with the same target in order to increase hit confidence, or, in contrast, if just one of the compounds annotated with the same target is active, this would then speak for a new mechanism of action. We will get to this point later again. You can also exclude compounds that modulate certain targets or target families to steer the type of biology that you want to discover. For example, away from kinase inhibition pathways. In practice, to verify your assay system, a curated compound set to interrogate, for example, hit-to-noise ratios, and also to check the validity of your system, known modulators of that phenotype are often pre-tested. The second scenario would be to compile a compound set that is intended to discover new targets. This is often referred to as chemical diversity sets or phenotypic-oriented libraries. These compounds may or may not have some knowledge about targets they hit associated with them. Referring back to the limits of assay throughput, you would still try to maximize the structural diversity within your compound set as best as possible. Factors like simplicity and complexity of the compounds can play a role in the follow-up of the screen, and compound sets can be designed using various criteria, for example, including privileged scaffolds or similarity cluster distribution within the set. Another point to make is that depending on your knowledge about the pathways leading to the phenotype, you can actually create bias by chemoinformatics and include compounds that should modulate pathways relevant to your desired phenotype. This is also sometimes referred to as hypothesis-driven library design. The current trend, however, is to start with small diversity sets and follow up with similarity iterations. So now that we have looked at the aspects of what system to screen in and the question of what compounds to use and the consequences out of that, we would now come to the point that we need to discuss on how to validate the hits coming out of those screens. Hit validation is a crucial aspect in phenotypic drug discovery, and it is advised to use a combination of methods to triage your hit sets. The most important ones are summarized here, and the general concept is noted along with it. We will discuss them in the subsequent part of this presentation in detail. For some, we will just highlight some key aspects. So we're going to look at counter screens, where you would screen against effects that are undesirable and could have interfered with the original assay and your readout. We're going to look at orthogonal screens, where you try to generate a comparable phenotype, but in a different assay to confirm your data. You're going to see how molecular phenotyping is used to detect and characterize the desired phenotype on a molecular level. And also we're going to touch briefly chemoinformatics, where computational force is used to analyze large data sets for structure-activity relationships, for example, and to correlate to complex biological readouts. Finally, we're also going to touch briefly on in vivo confirmation, where you would try to confirm a phenotype in an in vivo model if such a one is available. Counter screens are a cornerstone of every triaging cascade in phenotypic screening, same as for target-based drug discovery, to identify false positives and to de-risk the hit set. A classic counter screen is assay specific, meaning that you use the same assay and its readout, but with a different stimulus. This can also include the non-stimulated system with test compounds. Another common way is to perform counter screen by SAR, which entails testing chemically close analogs to substantiate the original hit finding data. Depending on the assay system and on the phenotypic readout that is used, often you would check for cell toxicity in different ways. The importance of cell toxicity testing is also illustrated here on the right. If your suspected mechanism of action is sort of a downregulation of a protein or a killing event of cells, any toxic interference as well as proper modulation of the pathway could lead to this phenotype and so produce false positives. Hits can also be further profiled against electron transport chain targets, which can also lead to false positive readouts. Lastly, one can directly profile questionable hits or hit clusters against known but unwanted targets or target classes to exclude those unwanted mechanisms of action. This will also be further reviewed in the section of the webinar where we discuss the elucidation of the mode of action. The next relevant item in the proper triaging cascade is an orthogonal screen to further verify the observed phenotypic effect to be real. In general, this is to show the consistency of the phenotype in two relevant assays. This can be, and you see this in the box on the right, A, the same cell but using a different readout, for example, gene regulation versus a functional readout, or, as example B, could be the same type of readout but in a different cell line, for example, a more advanced cell line like a human primary cell line is often used here after a recombinant one for the initial screen. If your hit set contains another compound annotated with the same target, that is from a different chemotype but shows no activity, this supports the fact that the annotated target might not be relevant as the actual mechanism of action. But be advised, ideally you would look into even further different compound classes to not base a mechanism of action devalidation on a single inactive. The following section will discuss molecular phenotyping, chemoinformatics, and in vivo model approaches to validate your hits further. Molecular phenotyping is a broad description of different techniques used to detect and to confirm a desirable phenotype. Additionally, these types of approaches also are used in mechanism of action elucidation, and this is discussed again later in this webinar. In the context of hit validation, molecular phenotyping means to compare the molecular state of the system treated with the drug with the untreated system, and this can be especially relevant to compare to the diseased or native system. That way, it can be confirmed or supported at least that the observed effect is actually the one that is supposedly desired and not an unspecific phenotypic readout. That was not the result of the desired changes on molecular level in case they are somewhat known. Now, what is the molecular level in this case? That, generally speaking, is a view onto the system's gene expression pattern, which could manifest, for example, in an up or down regulation of certain genes along with the treatment. But it can also be an analysis of changes in protein expression. As shown here on the right, often these effects are visualized in heat map formats indicating an up or down regulation versus the untreated system. Due to the slightly higher complexity of such an approach, this is often used in secondary or downstream validation of the hits. We are going to mention chemoinformatic approaches briefly here. Although this is a highly relevant and growing area in the context of hit validation of phenotypic screening hits, we can only refer to some very basics out there for the sake of time and complexity. The bottom line is that chemoinformatics can and have to support data analysis. In the field of hit validation, this is especially true since more and more modern high-content screening technologies are applied, generating a vast amount of complex data that needs to be properly exploited and analyzed. This is where chemoinformatics steps in and helps analyzing and visualizing the data so that useful conclusions can be made. With this, for example, an analysis of multiple endpoints can be performed to gain resolution on how compounds affect several biological aspects at the same time. Principal component analysis, or PCA, is such a multivariate analysis. Basically, the idea behind it is to reduce the dimensionality of a data set in which there are a large number of interrelated variables, while retaining as much possible of the variation present in the data set. Further, non-exhaustive examples of chemoinformatic analysis would be classification methods like tree decision models, random forests, and neural networks. For these neural networks, models are built to optimize the correct classification of the training set and then test data is applied to that network. In the example in the box, several cell aspects are analyzed, and the network is then interrogated if a cell, for example, is still healthy or cancerous. We are going to see more chemoinformatic topics later in the mechanism of action elucidation part of this webinar. The last item that we would like to bring to your attention in this first part of the webinar will be the question if an early in vivo experiment can confirm a phenotype that has been generated on a molecular level. As a side note, in vivo phenotyping can also be used for identification, not just for validation of hits in some rare cases. The idea behind the in vivo confirmation is similar to target-based drug discovery. It is to verify if the in vitro phenotype is actually linked to the desired outcome in vivo. While we acknowledge that this is often not straightforward to have a suitable in vivo model available, the benefit of looking into this is beyond the just mentioned confirmation, but that you also try to provide a safety assessment of the mechanism of action as early as possible. Needless to say, any compounds progressed into such a model need to have suitable pharmacokinetic properties, so that the pharmacodynamic effect can reliably be generated and attributed to the compound's target coverage. Looking back at what we have discussed in the assay section of the webinar, this early in vivo evaluation might be less important if you already have employed an assay system with high biological relevance and potential for high clinical translatability. You will find an example for an in vivo experiment for spinal muscular atrophy in the case study section of this webinar. Now, this concludes the first half of the webinar on best practices for phenotypic screening. In the second part, we're going to look into the remaining major topic of phenotypic drug discovery, that is the elucidation of mode of action, also known as target identification or deconvolution. Stay tuned.